Okay, so here is a mind map on the sources of variation for Year 12 biology. Here you have a chance now to take the full screenshot if you want to, but I'm about to zoom in and show you the various parts in a little bit more detail. All right, so if we zoom in, we can see up the top here, we've got point mutations starting off. And a point mutation is where only one base is changed. And this occurs either during DNA replication or exposure to mutagens. Okay, when we change one base, it's called a substitution point mutation. And when we either add or delete a base, it's called a frame shift point mutation. The effects of these are we got a silent point mutation where the amino acid doesn't actually change due to the degenerative nature of the DNA code. We can get a missense point mutation where a different amino acid is put into the polypeptide, or we can get a nonsense point mutation, stop codon is created. We can also get chromosomal mutations. Chromosomal mutations occur normally in meiosis, but can also occur due to mutagens. And they normally occur during anaphase or prophase 1. The effects of them, there are a number of types. There are deletion, where a section is lost, inversion, which means a section is flipped upside down and reinserted. Duplication means it's copied and then reinserted. Insertion means it's inserted into a different chromosome. Translocation means it's inserted into or exchanged with another homologous chromosome somewhere. Doesn't have to be the same homologous pair. And all of these affect the homeobox, which can result in infertility and cancer. Next, we'll pop up to crossing over, which happens in prophase 1, where the sister chromatids do not overlap. It is the non-sister chromatids. They cross over at a place called the chiasma. Okay, and when they do that, they swap information and become recombined chromosomes. This creates a new blend of the allele characteristics. All right, next we have independent assortment. Now, when the chromosomes assort, we say they move from the equator and they move towards the poles. In anaphase 1, the recombined chromosomes move to the opposite poles, and in anaphase 2, the chromatids of the recombined chromosomes, the chromatids move to the opposite poles. Independent assortment just says that there is no pattern, there is no set pattern as to which of the half of the chromosomes or the chromatids go to which pole. Okay, and that introduces a lot of variation. Now, when the chromosomes are pulling apart, if they don't pull apart properly, we call it non-disjunction. Okay, so when the homologous pairs fail to separate properly, this can occur in both anaphase 1 and anaphase 2. When it does occur, we can result in something called aneuploidy. Aneuploidy we define as 2n, which is diploid, plus or minus 1 chromosome. Okay, and this can result in such conditions as Klinefelter syndrome, which is XXY, Turner syndrome, which is XO, or Down syndrome, which means you've got 3 21st chromosomes. And then here is a picture of non-disjunction. The last thing we're going to talk about is ploidy. Now, ploidy is the number of chromosome sets within a cell. To find out the ploidy, we can do a karyotype, and over here we can see a karyotype. Here we've got chromosome pairs 1, 2, 3, etc. Down here we have the sex chromosome, so that makes these ones up here the autosomes. Okay, gametes are said to be haploid. They only have one set of chromosomes. Most cells are diploid, which means they have 2 sets of chromosomes. But you can change the numbers of sets by using a chemical called colchicine that upsets the spindle formation in mitosis and results in all of the chromosomes being in one of the cells. Right? From such use of colchicine, we can get tetraploid and octoploid organisms. During fertilization, if we've got the union of a diploid cell and a haploid cell, being a diploid gamete and a haploid gamete, we can end up with a triploid organism like a banana. If here is the normal protein production for a diploid organism, by having just 4 copies, so twice the number, we end up with twice the proportion of the proteins produced, which economies may like. Gives you a bigger yield. Okay, so as we move out again, we see the whole mind map again, and thank you for listening. Goodbye.